option but to do the class last week, but I hope all of you have watched the recording and covered it up. And so today I will quickly explain to you the plan. Okay. Online people, can you hear me clearly? Yes, send me a response. Okay. Right. So, for online people, people who did not receive the tutes, right, I know we have a few remaining from the last few, I mean a bit more remaining from the previous tute, right. Cellular basis. Everyone good? Yeah. Okay, online people. So, for people who did not receive delivery, right, of the tutes, I am sending the tute PDF to the Zoom chat. Okay. So, at this point you should have received 2 tutes like this. Okay. So, I know we have some parts of the blue tute remaining to do, okay. But today according to my plan, we have covered everything that needs to be discussed in order to do this practical seminar. Okay. So, today it is the practical seminar day, right. So, you must have received 2 copies like this. Okay. So, this one, as I think I showed this to you last week, it is going to contain only the diagrams with that, right. So, once we have fill in the blanks in this tute, you are going to have a nice short note which is a supplement to your resource book. Okay. It is an addition to the resource book, right. Resource book is your sort of the law and it is everything you need to write in the paper, right. But since you all are not robots, right, I think we need to have a sort of a diagrammatic and as well as a sort of a smart way of learning the content in the resource book in a sort of a smart way, in a scientific way. In order to do that, you need some additional diagrams and additional some sort of information organization. That is what I sort of intend to do with this, sort of rearrange the information present in the resource book in a way that you can actually get the concepts into the mind. Okay. So that's this one. The second one is the one that we are going to need a lot today. That is containing, that's the tude containing your classified past paper questions from the entire second lesson. Then make it, you can see for almost 12 pages, right? It is all past paper questions from just what? 6 years. 6 years of past papers. This many questions were asked from the second lesson. This many questions were asked from the second lesson. Got it? So you can see the question weight of the second lesson, ne? Chemical and cellular basis on your paper, ne? Right? And I have included the 2025 paper MCQs only because the structure essay paper is not available yet. Okay, it is available, but by the time I was making the tude, it was not there, right? So structured essay questions of the 2025 paper is not there. Okay, 2019, 20, 21, 22, 23, 24. That is there. This tude contains all the classified questions from there. So what we are going to need a lot today is from page 12 onwards. That is the set of practicals. Okay, the set of practicals that you need to know from the second lesson, starting from the biological molecules. Then, so under biological molecules we have few practicals. Then under microscope we have a practical. Then under enzymes we have practical, and photosynthesis and respiration we have practical. Those are going to come later. Okay, so that's the introduction to the tudes. Online people? Yeah, for now I only sent the question tude to the chat because that's the one we are going to need today. Right. Then the structure of the today's seminar, right? So I planned it as a seminar, no? Right? It's going to be structured like this. Okay? Let's see with time the maximum amount of practicals we can complete. Okay, right? So here we have two main sort of parts to this seminar. First one is the identification of biological molecules. That is going to be practical number. Okay, come to this tube. Make it. Okay, blue tube එක අදට අමතක කරන්න. එයා පොඩ්ඩකට අයින් කරන්න. Next week we are going to completely finish him. Okay, අදට come to the new tubes, page number 12. Okay, practicals, practical experiment 1, 2, 3, 4 and 5. They are parts of our first part of the practical seminar, right? So identification of biological molecules, right? That is the first part. So under that we have the first practical. What is it? What is the first practical that we have? Reducing sugars. Okay, reducing sugars. Number 2, very closely related to one, non-reducing sugars. Okay, reducing sugars, non-reducing sugars. Third one, starch, very simple one. Okay, identification of starch. Right. Fourth one, what is it? Identification of lipids. Fifth one, identification of proteins. So you know the only organic molecule that we haven't completely finished are the nucleic acids. So nucleic acids identification practical එකක් නැහැ, right? So with the knowledge you have currently, we can do all these practicals. Right? So that's first part. The second part of the today's seminar is microscope. Okay. Under microscope, you have two practicals, that is identification of the parts and functions of the light microscope and use of light microscope to observe specimens. So, that is the part 2. Okay. So, it is going to be, I mean, I have most of the things to perform all these practicals, and I brought a microscope as well. Okay. Do not ask me how I got it, right. So, this is the first part. Part 2 is simply microscope. Okay. Simply microscope. May take up the curve. Got it. With the time management, I plan to cover a maximum number of the practicals within the day. Right. So, every practical, this is going to be the structure. First, I am going to teach you the theory behind each practical, what is happening there, what is happening, I am going to explain in the board. Then we are going to go down, and at the table we are going to do the practicals. Okay. Then again we come and do the theory, we go do the practical, vice versa. Oh my, got it? Okay. So, any questions that you need clarifying? Online people, audio, video clear? You can see and hear me clearly? Yes? Yeah. No theory today, anyway. It's biology is a theoretical subject. So, without theory, there's no bio. Okay. So, the first practical is going to be from carbohydrates. Okay, it's going to be from carbohydrates. All right. What is the theory behind it? Identifying reducing sugars and non-reducing sugars. So, do you know the concept of reduction? Concept of reduction, don't know that. What happens when you are oxidizing or reducing something? So, this practical is the practical that is mostly questioned, most commonly questioned in your paper. I am going to explain these two together, right? Most commonly questioned practicals in the exam paper, right? So, I have to explain why I am taking so much effort. I am doing these practicals because historically there weren't much questions from these practicals, right? But sort of we are nearing the end of this syllabus, right? Within 2-3 years, this syllabus is going to be out. There's going to be a new one. So, towards the end, they have run out of things to include in the papers, right? Questions are all. So, at this point, it's going to be repetition. Situation. Because of that, there's a very likely chance that in upcoming papers, you are year 27 or something, the amount of questions asked from practicals is going to increase. Okay. So, that is why I am putting this effort. I did a similar seminar for my 2027 batch, and they took a lot of knowledge from that, and so that is why I am sort of putting this much time and effort into these practicals, right. Api balamu what's happening in a reducing sugar and non-reducing sugar sort of in a chemical sort of a perspective, right. What is the chemistry of it? Okay. I have explained right. So, imagine there's a certain species molecule substance is there. Okay. If that thing is giving an electron, is releasing an electron, what do you call that? If he is releasing an electron, what do you call that? He is becoming oxidized. Oxidation. He is undergoing oxidation. Okay, is undergoing oxidation. Then I always told you in this world there are no one is doing something alone. No one is doing something alone. Okay. If this person is giving an electron, that is because this blue color person is asking for an electron. නිකන් ජොලියට ඉන්න ගමන් ආමෙන් ඉලෙක්ට්‍රෝන් එකක් කියලා දෙන්නේ නැහැ. Okay. This one, this black species is giving an electron because this blue species is asking for an electron. මෙහෙම කියන්නත් පුළුවන්. This blue one is accepting this electron because this black one is giving the electron. Yeah, got it? So always for a oxidation reduction relationship, you have to have two species. Got it? You have to have two species. One is undergoing the oxidation. The other one, this one, who is accepting the electron, is undergoing reduction. Is accepting in the electron, is undergoing reduction. Okay. So you can say, okay, I'll say he is undergoing oxidation. Is undergoing oxidation, the other one is undergoing reduction. So that's not the word that we have here. මොනවද මෙතන තියෙන්නේ? Reducing, not reduction නේද? Okay. So how do you make sense of that? How do you relate that to this concept? Okay. So you can say, because this person is undergoing oxidation, he is causing reduction in him. He is causing reduction in him. Mu electron එක දෙන හින්දානේ මෙයා ගන්න. That's why he becomes reduced. So you can call him the person who is undergoing oxidation, or he is what? He is a what? Reducing. He is reducing him. He is a reducing agent. He is a reducing agent. Agent is someone who is doing something, no? Right? A secret agent or someone, right? He is doing a job, no? He is performing a task. What is the task of this agent? Reducing someone else. Okay? Reducing someone else. Right? So whatever. Then මෙතන කියන්නේ sugar. A sugar who is a reducing sugar, what is actually happening to him? එයාට මොකක්ද ඇත්තටම වෙන්නේ? What is actually happening to him? He is undergoing oxidation. That's why he is a reducing. He is reducing someone else. Okay? Right. So let's focus on this one first. Our identification of reducing sugars. Okay? Identification of reducing sugars. So how do we do this practical? How do we do this practical? Okay? The reagent, right? Onama chemical, onama sort of a reaction in biology, we use a preformed chemical, right? What we call as a reagent. Okay? So it's a preformed sort of a chemical substance that we can use to sort of get a sort of a textbook result එකක් ගන්න පුළුවන් using a certain reagent. If you follow these steps, you should get a sort of a well documented result. මේ වගේ එන්න ඕනේ කියලා තමයි, that is the theory. Okay? So the test that we use to identify a reducing sugar is the Benedict's test done using Benedict's reagent. Okay? So we call it the Benedict's test. Okay? Benedict's test. So what do you have in the Benedict's reagent? Benedict's reagent, okay, what do you think you have? Something that should react with a sugar that is giving electron. Something that should react with the sugar that is giving an electron. This is the Benedict's reagent. Okay? Online people? You can see, right? Slightly reflecting, I know, right? Yeah, I'll try to write in bigger. This is the Benedict's reagent, right? What color is it? Blue. right? So, using basic chemistry, what sort of species have a blue color? What sort of a species have blue color in chemistry? Samanning, what is the, okay, what is the metal ion that has a blue color in aqueous solution? Copper 2 plus, right? Copper 2 plus, Cu2 plus aqueous has a blue color. හරිද? දන්නවනේ? your Benedict's agent කියලා කියන්නේ aqueous copper, I mean copper 2 plus in a alkaline medium. Alkaline කියන්නේ slightly basic aqueous copper 2 plus solution. Right? Basic copper 2 plus solution. Simply that. Okay. So what we are going to do is we are going to react it with a what? We are going to react it with a reducing sugar. Copper 2 plus api when you react it with a reducing sugar, what's going to happen? Yeah, this reducing sugar is going to give this copper 2 plus an electron. Copper 2 plus an electron. Okay. So then what's going to happen is you are going to have copper plus. Yeah, copper, this is a reducing sugar. He is undergoing what? Oxidation. Or you can say he is causing reduction in him. He is making him reduce. So copper 2 plus is going to become copper plus. This is cuprous, ne? Again cuprous, copper plus. Okay, copper plus Q. This copper plus is very unstable. Okay, copper 2 plus, okay, he is not going to remain for a long time in a solution. Or විදියට දිගටම ඉන්නේ. දැන් මේ Benedict solution එක ඉන්නේ පාඩුවේ, right? Copper 2 plus is stable in aqueous solution. But your copper plus is not like that. What's going to happen is he is going to react with oxygen. He is going to react with our atmospheric oxygen O2. Okay, it's going to produce a oxide Cu2O. Okay, is going to produce an oxide that is called Cu2O, cuprous oxide. Okay, this cuprous oxide is a solid. Okay, it's a solid. So when something solid forms in a solution, what do you call that? It settles down. Settles down. What do you call that? A precipitate. It becomes a sort of a sediment that is going to settle down in the solution. Okay. Okay, then the reducing sugar is going to give an electron that is going to reduce the copper 2 plus into copper plus. That copper plus is going to react with oxygen to produce this precipitate, which we call as having a brick red color. Okay, brick red color. Brick red color. හැබැයි this doesn't form directly. This brick red color doesn't arrive whenever we put a reducing sugar, the entire copper 2 plus doesn't convert into copper plus immediately. Okay, it undergoes several transformations. It undergoes several sort of transition. I don't know exactly the full chemistry behind it. Okay, ask Chamudita sir, right? Okay, so it undergoes some sort of in between metal ion complexes that are going to have some several colors, right? So those colors we can observe today. First you have blue. Blue color is going to become, this blue color is going to become green. Okay, it's going to become green. Okay, then it's going to become yellow. Okay, so yellow, I don't have a yellow pen. Okay, yellow, orange. Okay, then you are going to have a red solution, right? Red solution na kambiro. Okay, red solution. Then you are going to have a brick red precipitate. Got it? This is the sort of the color, let's say, transition from this initial blue color at the end to the brick red precipitate. Okay. Onokai Benedict's test එකේ. So that's the theory. Okay. That's the theory. Wherever you go, I mean, so imagine you progress from A levels to maybe medicine or something, right? In the first 2 years, we have a subject called biochemistry. Right? Biochemistry is a subject. First 1.5 years, we have only 3 subjects, no? Anatomy, biochemistry and physiology. Okay. At the end, in our faculty, we have an exam after 1.5 years that is called the first MBBS exam, right? Then we have another one at the end of the 4th year called the second MBBS, then it's the final MBBS at the end of the 5th year. Right? So first MBBS is a bit serious business in our faculty, because why? If you don't pass it, so you can fail it for one time, okay? Then you have to rewrite it. If you fail it for a second time, you will be demoted to the below batch. Batch miss karna agila. Okay? You will be put in the below batch. So imagine you went to the university, so api samane vaten 2028 intake kila, right? You will have to finish your degree with the 2029 intake, okay? If you repeated in the first MBBS in our faculty, batch miss karna, right? So in the final sort of the biochemistry exam in this first MBBS, we have a written paper and a viva where we have to sort of face a panel and they are going to ask questions in real life. Then we have a sort of a projected questions, right? Project questions, and within 1 minute we have to write answers. That is called an OSPI. And finally, we are going to have a practical session exam for this biochemistry subject. So in this practical session, what we have to do is we have to do some tests. We have to identify reducing sugars, we have to identify proteins in it, we have to identify bile salts, right? Yeah, that's basic. And ketone bodies, diabetes, there's another chemical. Identify. What do you think we test for? In the exam, what are we given? Then for today, if we are doing the reducing sugar test, I'm just going to take some glucose powder and going to make a solution and do the practical. What do you think we are given in that exam to do this test? To test for sugars and proteins, bile salts, or what do you think? Urine hambit. Okay? Online people, what do you have to test for in a biochemistry exam? Yeah, urine. So how do you get urine? How do you get urine? Someone has to donate it, right? For any practical, I'm just going to give you the basics into any sort of a practical examination. How is the own practical examination? Okay? Ekka experiment ekak kalla madi. Okay? If you want to prove something, okay, I got this test positive. I gave you a reducing sugar sample, and if you want to say, okay, this is positive, you have to have a test that is negative, right? Any test you have, I mean, any experiment you are doing, you should have something called as a control experiment. Control experiment, right? So imagine you have a test tube with the reducing sugar in this. You have to have a sort of a, imagine maybe it contains similar volume of water that you are going to do the Benedict's test. Okay. Something that doesn't have the whatever you are testing for, right? So on a practical level, you should have a what we call as the experiment, or you can say the sample and the control. Okay, the sample and the control. Then imagine you are testing for sugar in a urine sample. Okay, the urine test right? Okay, imagine you are testing for urine in a sugar in a urine sample. How do you get a urine sample with glucose? Glucose skeleton. Again, someone with diabetes. Someone with diabetes. Do you know then sort of in traditional medicine, do you know how these sort of traditional doctors, Ayurvedic or others, do you know how did they diagnose diabetes? Diabetes diagnose kare ko they will. Did they have glucometers? Then you know blood drop pack karam meter dala, they are going to tell you the amount of blood sugar earlier? No. How did these traditional doctors identify diabetes? Huh? Yeah, then no. What? Do you urinate? To a? Just urinate to the ground. කොහේ ගහක මුලට ගිහිල්ලා urinate කරන්න කියනවා. Right? Just urinate on the ground, somewhere outside. Okay? What's going to happen? Within a few minutes, ants are going to come there. කූඹි වහනවා to the urine. If it's a severe diabetic patient. Why? They are going to have a lot of glucose in their urine. Why does a person with diabetes have a lot of glucose? One thing, in their blood they have a excessive amount of glucose, so that's going to come out filtered from the kidneys. Okay, so people with diabetes, you can get us so many urine samples from our teaching hospital, right? එදා උදේට hospital එකේ අර bed pans තියෙනවානේ, sort of catheter bags, urine bags එකලා තියෙනවානේ, ඔයයින් collect කරලා ගේනවා. Okay, patient's urine. Then ඒ වගේම people with kidney disease, right? Of course, people with diabetes are going to later develop kidney disease as well. What's going to happen is your kidneys are like a very sensitive filter. චූටි හිල් තියෙන very small holes තියෙන filter එකක් වගේ. Okay? With a long time diabetes or sometimes your kidney disease, what's going to happen is those small holes are going to become big with time. Okay? So imagine you have a filter with big holes. ඔක්කොම ටික යනවා ඒක තුරට. So your proteins in the blood are not supposed to go into the urine. Right? කන ටික එහෙම යනවා. Right? That's why diabetic people get thin. Get වෙනවා කියන්නේ. දියවැඩියවද කියලා අහනවානේ, someone is getting very thin. Right? That's because with a long time diabetes, they are going to lose proteins in the urine. Okay? So people with diabetes can have proteins in their urine as well. If we tested you, you are not going to have urine or glucose in your blood, in your urine. Okay? So then hurry. From a kidney patient or a diabetic patient, you can get protein containing urine as well. Then make control. How do you obtain the control? Again, a normal urine. Yeah. On the morning of the practical, all the boys in the batch should go to the Department of Biochemistry to donate urine, because to use as the control experiment. Okay. Now, the batch again, we have only about 180, so only about 65 are boys. Okay. All the others are girls. Right? So there's a limited amount of urine that we can donate. So for one person, at least 20 to 25 milliliters of then urine. Okay. So that's going to be a lot of urine demand, right? So all the boys are told to drink a lot of water in the night and come early morning to the department. In the washroom, there's a sort of a 5-liter glass beaker on the toilet, right? You are supposed to, right? We are going in the morning. We have an exam just after, but we have to hold it until we just before the exam. Urine, right? So we have to make that sacrifice anyway. So control. Or randomly, batch diabetic control positive, right? No, that's not enough. Okay. So usually also there's not enough urine to be used for the sample as well. In diabetic patient, right? Even in Kalubovila hospital, not much. So what they do is artificially they add some glucose or they add some protein from outside so that we can get a positive result. Okay. So that's what sort of a applied experiment is like. When you sort of apply the theory in a real life situation to see if a person is diabetic or not. That's the real life application that we are sort of preparing you for. Okay? The medical faculty gilla already karanno. Okay. Hari. So reducing sugar and the non-reducing sugar, both concepts I'll explain right now. Okay. So reducing sugar, no issue. A reducing sugar can give an electron to your copper in the Benedicts so that you can get this color change and the precipitate. Hari. What's going to happen if it's a non-reducing sugar? So who is the non-reducing sugar that you know in your syllabus? There are only 6 sugars that you know. Sugars higher than, yeah, 3 monosaccharides and 3 disaccharides. All monosaccharides are what? Can. All monosaccharides are reducing. Most of the disaccharides are reducing. Who is the only non-reducing disaccharide that you have? Sucrose. Sucrose. So we are going to test for sucrose today. So how we test for sucrose is basically that same experiment. Right? What we are going to do is, we are going to convert this non-reducing sucrose to a reducing sugar. That's what we are going to do. So, may experiment ek matamai. What we are going to do is, we are going to this sucrose, we are going to add an acid in aqueous HCl. Then we are going to heat it. Okay, we are going to apply some heat. Okay, then what's going to happen is, so how do you make a disaccharide? Abhi disaccharide hadanne komada for monosaccharide? By a dehydration reaction, losing water. Ne? So if you want to break a disaccharide, how do you do that? If you want to make a disaccharide, you take water out. If you want to break one back into monosaccharides, you add water. Right? You add water. How do you add water? Using an aqueous acid. Right? Organic chemistry wala this hydrolyzation, you will learn so many reactions. මොකද ගන්න? Aqueous dilute acid එකක් දාලා heat කරාම හරි. Water molecule gets added. Okay, so when you do this, what are you going to get? What are the monosaccharides of sucrose? क्या? Glucose and fructose. You are going to get glucose and fructose. Then hurry. Glucose and fructose are both reducing sugars. Then again, we are going to do the Benedict's test for this. We are going to do the Benedict's test for, as the next step, we are going to do the Benedict's test. Then you are going to get the same color change. Okay. But if you do just the Benedict test, dongala sucrose, if you just do the Benedict's test, you are not going to get any color change. Because why? Sucrose is a non-reducing sugar. Okay. Clear? This is the theory of the practical. Okay. So, come to the practical guide. Page number 12, identifying reducing sugars in the laboratory. Okay. Reducing sugars such as monosaccharides and some disaccharides, right? Highlight what is the list of your reducing sugars: glucose, fructose, galactose, maltose, lactose. Highlight the list. Highlight the list. Okay, can be easily identified in the laboratory using Benedict's test. Okay. Right. So, I want to give you sort of a idea of this reducing nature. Abhi wenna metana. There are some space no practicals kiyana thana podda api. Let's try to write what is this reducing agent, reducing sugar and not. Okay. Right? There is some space no. Me yata liyanne epa because we have to draw some practical steps there. Mena me patte wage, right? Somewhere you have space. Okay. Got it? Let's write a reducing sugar, a reducing sugar. Let's write a reducing sugar, a reducing sugar is able to, a reducing sugar is able to donate electrons. A reducing sugar is able to donate electrons. A reducing sugar is able to donate electrons and reduce another species, is able to donate electrons and reduce another species while undergoing oxidization itself, while undergoing oxidization itself, while undergoing oxidization itself, while undergoing oxidization itself. If you like, you can write down that reducing agent undergoes oxidation, it gives an electron, the other species undergoes reduction. Chutti diagram me akkandi, right? Chutti diagram me akkandi. The reducing agent gives an electron. Okay, Chutti diagram me. Only that one, right? Benedict's test gave us sucrose eva unnai. Got it? Okay. Then the procedure of Benedict's sugar is like this. Let's read what is the procedure of the Benedict sugar. That's what we are going to do, right? What is the procedure of Benedict's test? Let's read. What again? It's a Benedict solution is a mixture of copper sulfate and sodium hydroxide. Copper sulfate tamai how you get the aqueous copper 2 plus. Reducing sugars reduce soluble blue copper sulfate containing copper 2 plus into insoluble copper oxide, insoluble brick red copper oxide containing copper plus. Okay, highlight that. Copper 2 plus, blue copper 2 plus to insoluble brick red copper oxide with copper plus. Pulvanam blue and red color will underline karanna. Okay. Yes, I sent a tute PDF to the group. Sorry, in the Zoom chat. I'll send again. Okay. So what is the procedure? Well, again, add 2 milliliters of solution of reducing sugar into a test tube at an equal volume of Benedict solution. Shake well and boil in a water bath. Initial blue color changes to a brick red precipitate. Simple. Okay. What is the procedure? Right. Let's talk about the non-reducing one later again. Okay. How do you do this? into a test tube, you add. So the reducing sugar we are going to take here is glucose aqueous 2 milliliters, the thing, 2 ml. Okay. Then we are going to add to this. To this we are going to add Benedict solution equal volume. So that is going to make the solution blue. Okay. 2 ml Benedict. Okay. Then what's going to happen? We are going to heat this until it boils. We are going to heat this until it boils. It's going to undergo that color changes, green, yellow, orange, red, and finally you are going to get a, at the bottom, you are going to get a brick red precipitate. You are going to get a brick red precipitate. Okay, the solution is there. Aqueous solution, right? In that solution, you are going to get a precipitate. Okay, this is the practical. Okay, so let's try this. Try right. So for this, so yeah, first trying out the reducing sugar test. Okay, so this is the Benedict solution. That is, Benedict solution is the main reagent that you are going to need. Okay, then what else do you need? In order to heat everything, you are going to need some boiling tubes. Okay, and to hold the boiling tube, you are going to need a test tube holder. Okay, and to heat everything, because mata genda bෑන මෙතෙන්ට Bunsen burner එක, right? So I'm going to use a sort of a small spirit lamp. Okay, then this is a sort of a high tech pipette filler that I can use to suck liquids into, so that I can inject them into, so not inject, sort of use them in the experiment. Okay, so first we are going to need to make a glucose solution. Right, so we'll make a small glucose solution. We have to try to make it saturated. Okay, yes, it's there. Can you see? There's a row here. You can count. Yeah, at the back. You can't see. So Negombo got the glucose, so it's not open yet, right? Okay, so your glucose is going to be the reducing sugar. It's the sort of the textbook test that we can do. Okay, so you have to have a nice saturated solution, because glucose is the one who is giving the electrons, right? If you don't have enough glucose, it's not going to give a good positive, good enough result. Okay, so I didn't have a glass rod at home, so I just brought a straw. How is it? Online people, can you see? Yeah. Okay, so we have a glucose solution. Then what are we going to do? We are going to add Benedict to it, equal volume, and then we are going to heat it until it boils. Okay. Fire alarms, right? Okay. So these Benedict solutions, as you know, they are aqueous solutions, and once if they have been exposed to the atmosphere for some time, they are sort of the amount of ions that they have can reduce. Okay, so our volume පොඩ්ඩක් බලාපොරොත්තු අඩු කරලා පටන් ගත්තා, right? Very unlikely that we'll get a precipitate. Okay, really unlikely we'll get a precipitate, but there will be a color change, right? We'll see if there is a color change. Okay. So to this boiling tube, we'll add some of our glucose solution. No need to do much measuring, right? Equal volume add karana hari of what? Our Benedict solution, right? We add Benedict and then we heat. So equal volume when, okay? Now people, can you see? So you can see the color of the Benedict, blue color. The pipette is very small, right? I have to use it several times. Is it twice? Okay. It doesn't have to be 2 milliliters, 2 milliliters. It just has to be equal volumes. Okay, equal volume. So you hold it with the test tube holder, and then you heat it. Always basic sort of lab safeties you have to. So Hari, Mulima, what is the color? You can see it's blue colored, no? Right. That is because of the aqueous copper 2 plus. So let's see what's going to be the color. Always you have to heat everything away from your face. Okay, you have to heat everything away from your face. So why do they say to use a water bath? To give some heat evenly, and also sometimes there's a risk of the boiling tube sort of bursting, shattering because of the heat. But this is a borosilicate one, so hopefully it won't. Okay, because of the AC, the flame is you have to heat it until it boils. Huh? Okay, so a Bunsen burner is able to provide a lot more heat because it's gas, no? Right? You can see a small color change, right? Tikak greenish way, right? Slightly becoming greenish. Can you see? I'm putting a teal color, sort of green blue, ne? Yeah. Now green, no? There's a AC coming from this side. Yeah. So we are sort of almost all green now. So now we are getting close to yellow, right? Ticking yellow and all people, can you see? Sort of a muddy pond water again. Sort of, let's see what is the end color we can get, right? Red benagam ya idu balu. Sort of a diarrhea yellow karak thine. So we are getting close to a bit of orange shade now. Bit of orange. Online people, can you see? Send me a response. See? Orange. I don't think it will go any further because the glucose nowadays, it's not pure glucose. Vitamin C so it's not purely glucose. So you have a nice orange color. I think that's all the color change we are going to get. It didn't boil yet. That's the problem with the ASEAN. It's a small spirit lamp. Boil ka namarui. I think we have to be satisfied with that. Either orange. But what should be the ideal result? A brick red precipitate which is copper 2 plus, right? Sorry, copper 2 oxide, right? Yeah. Okay. Right. You can see, right? So what is the conclusion from that? The copper 2 plus in the Benedict was reduced to copper plus by the glucose, which is a reducing sugar. Okay, simple as that. That correct? Okay. Right. So Megam api, let's do the second experiment as well, which is non-reducing sugars. Right? How do you identify non-reducing sugars? What do you have to do to it? What do you have to do to identify non-reducing sugars? You have to take a non-reducing sugar and convert it to a reducing sugar. Take a non-reducing one and convert it to a reducing sugar. Yeah. So what is a non-reducing sugar that we are going to use? Yeah. What is a non-reducing sugar that we are going to use? Some sucrose. Okay. Bit of sucrose we are going to use. Okay. So we don't need the glucose solution anymore. Oh yeah, I have 3 beakers. So in order to do this, what needs to be done? A practical guide diagram. Right? What needs to be done here to identify non-reducing sugars? Yeah. So you take a non-reducing sugar and you add an acid and then you heat it. Okay. You add an acid and then you heat it. Then the solution becomes acidic. The solution becomes acidic. Identification of non-reducing sugars. Right? We'll write a note. Okay. Method: add 2 milliliters of non-reducing sugar sucrose to a test tube. Okay, non-reducing sugar sucrose to a test tube. Add 1 milliliter of dilute HCl. Right? Dilute HCl, that's how you acidify. Then you boil for 1 minute. The sucrose is broken down to the to the monosaccharide parts. Glucose and fructose. Carefully neutralize with sodium bicarbonate. Okay. Hydrogen carbonate. Check with the pH paper. pH paper okay. Add an equal volume of Benedict solution. Shake well and boil in a water bath. Initial blue color changes to brick red precipitate. Same story. Okay. So that's what we are going to do now. So to do that, we are going to need a what? To make a sucrose solution. Okay. Right. So to make the sucrose solution, exactly same procedure as earlier. Okay. So you take some of your normal sucrose. I mean, then they say to take normal white sugar because it's been refined, right? So if you take your brown sugar has certain sort of other organic things, different taste in brown sugar. That is because it's not been refined. So it has some other sort of organic substances from the sugarcane plant. So white sugar is mainly basically all sucrose. Okay. So you make a sucrose solution. You have to have a saturated solution because in the cold water. Okay. I will keep it for some time to dissolve. Okay. We will keep it to get dissolved because we are going to need a very nice saturated solution. So how do you know it's a saturated solution? Then a saturated solution there should be some of the sugar that is not dissolving at the bottom. Again, dissolve. There's no more space among the water for the sucrose to go. Right? That means it's saturated. So almost I think saturated. Then what else do you need? You need a acid, right? So acids are very hard to find. So I have only a small amount of acid. This is dilute HCl, right? It's not exactly dilute, it's 5m, okay, right? Concentrated, right? Change the chuttadam, okay. Let's try to resolve this even more. Okay. So protein test. All right. Any questions? Okay. So we'll take this test tube, right? A small amount of sucrose. Then what do you need to do? We need to acidify it, right? අපි ඒකට වෙනම pipette එකක් use කරමු, right? Few drops of HCl, okay? And you have to be careful not to suck it into the pipette itself, right? Okay. Let's add a little bit more, right? Let's add a little bit more. Okay, right. So we have added the acid. Then what do you need to have? You need to have someone to neutralize this. we need someone to neutralize this. So, to neutralize this, we are going to use sodium bicarbonate, but I couldn't find sodium bicarbonate, so we are going to use calcium bicarbonate, which is a bit weak, okay, which is a bit weaker than sodium bicarbonate, but let's see what happens, okay. So, we are going to need a aqueous solution of hariranang sodium bicarbonate, right, but let's try to make it work with calcium, okay. Right, so we will keep it to a side. What do we have to do next? We have to with the, so then this is sucrose with the acid, no? Sucrose with acid. What do we have to do to it? We have to heat it. Because why? Sucrose has to become digested. Sucrose has to become digested. So we have to heat it to convert the non-reducing sucrose to something reducing. So always in our labs, so girls, if you are when you are going inside, because we have Bunsen burners, there's a switch, no, a switch again, there can be the flame can you can make it become very big, right? So you always have to have your hair tied, right? Because hair konde giniganna very fast, okay? ගිනිගත්තොත් ඉතින් ආයි කරන්න දෙයක් නැහැ, right? කෙලිම fire extinguisher එකක් ගහන්න තමයි තියෙන්නේ, okay? Right? So all the girls should have their hair tied and before going to the lab, okay? So first day of the biochemistry lab bara nikan hena lecture එකක් දෙනවා, these are the basic safety principles, okay? And also in the lab we usually wear gloves and sort of eye protection, because we are using sort of infectious, urine can be infectious, blood we use, right? So those can be infectious, right? So we have to use gloves. So then methane hema nahi cutting got the glucose again, right? So nothing bad about it. We have to heat it until it boils, okay. I can see the sort of the smoke coming, so that should tuck hold it. So the sucrose should be, theoretically it should be digested, let's see, right? Because we are not exactly measuring the amount of acid, it may be enough or not enough, okay. I think that's enough, right? So we'll keep it to cool down a little bit, right? Should cool down another. So what's the next step? You have to neutralize it with the sodium bicarbonate. Then you add Benedict. When you do the Benedict test, you again heat it. Got it? Simple as that. So this is acidified, no? Right? Still it's a bit hot, right? Then today, if we have time, we have the lipids identification, and for that also I brought some honey also. Then Joliet wage karanna puluwan, these are not in the syllabus. This is a aqueous solution of honey, okay. The honey, if you do a experiment of reducing sugar or whatever, if you do a reducing sugar test, what's going to be the result? Honey will be nothing. Fructose, is it a reducing sugar or not? It is. So it should theoretically give a positive result. Habai, then how do you imagine if it was sini dala hadapu honey, sugar honey? Ema fake, right? If it had sucrose, is it going to be a reducing sugar positive? No. So we can try just for fun. We'll both try at the level. Okay. Then I think I have some fat-free milk also, right? Fat-free milk because it's going to mainly have lactose. Right? If you remove the fat-free, then without fat, it's going to be mainly sort of sugars considered, right? Proteins and lactose. So we can do the what test? Lactose now? Is it? It's a reducing sugar, right? Yeah, lactose. So lactose should be positive for the Benedict's test, right? Okay, if we have time, let's see if we can. And we have some coconut oil to do the identification of lipids, some egg white to do the identification of proteins, right? Those are the standard tests. We try at them okay? It's already 5. So we should neutralize this with the bicarbonate. Can you see it's a acid base reaction, Penadana, I can hear the sort of whizzing sound. It's acid base neutralization reaction, right? There's going to be what? Carbon dioxide gas. Okay, that's because the acid is a bit too concentrated. So ideally we should test it with a bit of pH paper. Let's see if it works. Okay. So this color will settle. Okay. So you can see it's a white color solution. Then what we are going to do is we are going to add Benedict's. We have to add Benedict's into this. Then we have to do the sort of the normal Benedict's test, right? The Benedict's solution is also alkaline. The Benedict's solution is also alkaline. So we'll see what effect it's going to have. Yeah, so the acid has been neutralized, right? Because of the milky color, podi issue but let's see. Okay. Because of the heat, can you see? The tube is already hot. It's becoming yellow. Got it? Online people, can you see? Sort of greenish colored already. That means what? The sucrose has been converted, right? There is some residual heat from the previous boiling. Can you see? Dana color right? So let's see what's going to happen. Hello people, can you see? Let's add a few more Benedict, right? Okay, right. Already we have a positive result, right? Yeah, we already have not the blue color of Benedict. There is already a yellow orange color. Without heat karam it's intensifying, more and more orange. Yeah, can see. So we have sort of a almost a bit closer because we use calcium podi milky color එකක් ඇවිල්ලා තියෙන්නේ, right? Because they don't give out strong acids and bases anymore, so very difficult to find. Okay. Even for scientific purposes. Got it? We have sort of a bit light because of the calcium bicarbonate we use. ඒක ගණන් ගන්න එපා. We usually have to use what? Sodium bicarbonate තමයි ideally දාන්න ඕනේ. But we have orange color. Right? Got it? Experiment එකේ sort of the third color change we have. Got it? Clear? We can't bring it to a boil because of the small spirit lamp. Boil එකකට ගේන්න අමාරුයි. So that's it. Okay. Those are the two reducing sugar and non-reducing sugar practicals. Got it? Online people, you can see this is the one with glucose, this is the one we did with sucrose. Okay. Right. Let's write a quick note. Okay. So there in page number 12 there's a space. Practical experiment 1, identification of reducing sugars. Okay. Draw this. Okay. You have a glucose solution 2 milliliters. Okay. Then you add 2 milliliters of Benedict, then you apply heat. You are going to have a brick red precipitate at the end. Then I want you to write down this. Copper 2 plus reacts with reducing sugar, it becomes copper plus. It's going to react with oxygen to become cuprous oxide, which is the precipitate. So draw this sequence of, write down this sequence of color changes from the blue, green, yellow, orange, red, and finally the brick red precipitate. Okay. Then what's going to happen in a non-reducing sugar? What's going to happen in a non-reducing sugar? Yeah. So in page number 13, that's what we did, right? 2 milliliters to a test tube, 1 ml of dilute HCl, boil for 1 minute, right? So let's draw that one. Make a complete, right? You have to draw this. This is the sequence of color change. Okay, this is the sequence of color change. Okay, done. So what is the procedure for identifying a non-reducing sugar? It's almost the same, but we'll draw a new one, right? So from here onwards, it's a bit difficult. Different, right? So yeah, there's a stage that you add 2 milliliters of Benedict and you heat it. But before that, what needs to be done? You take, yeah, this is 5 ml sucrose solution, yeah. Then you add what? About 1 ml HCl dilute, okay, about 1 ml dilute HCl, okay. Then what do? You heat this, you heat this. Then after heating, you add what? To neutralize, you add sodium bicarbonate aqueous, the volume given. No need. I mean, whatever you need to add to neutralize. pH paper can bala you neutralize, right? So you can say neutralize with sodium bicarbonate. Then you add Benedict. Then you heat. Then you are going to undergo this color change. What is the color change? Green, yellow, orange, right? You write down the full sequence and you are going to get this. Okay. So methanadi we don't actually say 2 milliliter because we have added other things. No, we have added other things to solution. Acid deka dala thiyena, sodium bicarbonate dala thiyena. So we usually say equal volume. Whatever you have here, you add equal volume Benedict. So write down there on page number 13, practical experiment number 2, manage your space, non-reducing sugar. This is the procedure. You make a 5 milliliter, sorry, it's 2 milliliter, right? I'm sorry. 2 milliliter solution of sucrose, add 1 milliliter of dilute HCl, you heat, then you neutralize it with sodium bicarbonate, aqueous, then you add equal volume of Benedict, then you heat, you can see the series of color changes. Got it? Okay, done. Done, right? So while you are drawing these, demo padang kan apo may practicals nang apo padang kan na tiyagan depa because you have a lot more many things to do in the sort of a lot more content to be covered, right? So to be memorized. So just done with these practical wage deval, be done with this right now. Okay. So what next? Mokad dilang rakan. Which practical do you want me to do next? According to the practical guide, what do you have next? Starch. Simple one, no? Identifying starch in the lab. Shall we do the protein test before? Abhi bioret tech kar le mudhe. Practical experiment 5. Dekha jolly talk 1 kar mudhe. Then we can take a break and do the rest. Yeah? Okay? So, identification of proteins, page number 14. Abhi nahi aayega karamu. Identification of proteins in the laboratory using bioret test. So, what happens in the bioret test? Bioret reagent was actually found to identify sort of for these sort of industrial chemical processes, but we adapted it to be used as a way of identifying not exactly proteins. So yes, we use it to identify proteins, right? So here actually what we, I mean the reaction that is happening is with the bioret reagent and peptide bonds. Peptide bonds, you know, connecting two amino acids, what do you have? Connecting two amino acids, what do you have? A peptide bond. Eh? So, second test for today, bioret test. Okay, bioret test. What we are detecting here is, you know, this is a amino acid, this is a amino acid. Connecting those two, you have a peptide bond. In this peptide bond, to be exact, there's a nitrogen with these two lone pairs of electrons, and there's another hydrogen, then there's a carbon with the double bond, and then carbon, maybe carbon. This is the peptide bond. Eh? These two electrons in the nitrogen is going to have a sort of a interaction with the species in the bioret reagent. So, what do you call as the bioret reagent? Maybe bioret reagent, let's read, okay. 5% KOH and 1% copper sulfate. Okay, 5% KOH and 1% copper sulfate is what we use to do the bioret test. So, what we are going to do is, what is the procedure? To a test tube, okay, to a test tube, you add some, well, again we have to use 2 ml egg albumin, yeah, egg white again, eh? So, egg albumin to a test tube, then what do you do? Okay, then you add equal volume of 5% equal volume 5% KOH or NaOH you add. Okay. Then what do you do? Then what do you do? 2 drops of 1% copper sulfate. Okay. 1% CuSO4. How many? 2 drops. Okay, 2 drops. 2 drops of copper sulfate. Then what are you going to observe? What are you going to observe? A violet or a purple color develops slowly. in this solution you are going to see a purple color, okay, violet or a purple color you are going to observe. So, I can indicate it a bit like this. Right, sort of purplish color. Okay. This proves the presence of what? Peptide bonds. That's all it shows. Okay. Biuret test shows the presence of peptide bonds. So, not only proteins, you can have a positive result for this. Is this a protein? Amino acids, two amino acids that are just connected by a peptide bond. What is it? It's just a dipeptide. Ne? It's not a protein. It's just a dipeptide. No? So, even a single dipeptide can give a positive result for the Biuret test. Okay. Then it's been easier for us. It's been made easier for us. Okay. It's been made easier for us because we don't need to do all these things. There's a pre-made Biuret reagent, right? Biuret reagent whenever you can buy. Okay. So, let's draw this. Okay. Then we can do the experiment. Okay. Practical experiment number 5. 2 milliliters of egg albumin, you add 5% NaOH or KOH, equal volume. Then you add 2 drops of 1% copper sulfate. You are going to get a color change, violet or purple. Violet or purple color. Simple one. You are detecting the presence of peptide bonds. Okay, peptide bonds are present in proteins. You are going to get a violet or purple color. This color change is the only thing you need to know. Okay, when you add Biuret reagent, you are going to get this color change. Okay. So, Biuret reagent, as I told you, we don't need to have this KOH and NOH aromeva. You can have a direct Biuret reagent pre-made, okay, for the detection of peptide bonds. We use this in industrial purposes, in sort of food processing, aromeva ganna Biuret reagent. Okay. So, we don't need any heating or anything. Okay. So, the heating experiments are sort of done. I mean, for fun, we can do a few more fructose aromeva. Okay. So, what we are going to do is do the exact experiment that they have given in the practical guide. This is egg white. Okay. Egg albumin. What is another name for egg albumin? Ovalbumin, right? Ovalbumin. So, we take a test tube. I think we can directly add, I mean, no issue, right? Add a bit of online people, done, right? So, we have some egg white, okay? So, you add it to a test tube. Godakawa, right? So, because it's sort of a viscous solution, right? Either you have it completely or not, right? It doesn't matter, right? Right? Because it has become a bit of a colloid, a coagulation, right? Egg white, it has coagulated a bit. I had to. I broke it at around 12 p.m. today, right? Kusira panla, okay. So we take some biuret reagent. Yeah. So we don't need to do all these elaborate things. KOH dala one few drops or we can directly add. Okay, you can already see a purple color. Okay, window, right? Okay. So some of the yolk has also come because I'm not that experienced. Right? Some of the sort of the yolk is not also there, but you can see the water soluble protein is at the bottom. So it's very purple colored already, right? So that indicates the presence of proteins. Okay, so online people, it's not my purple shirt. Okay, so you can mix this a bit. Okay, it has coagulated a bit, right? The white has coagulated a bit because of the heat and everything, right? Can you see? It's exactly the same color, right? Okay, got it? So this is the positive biuret test. Yeah, can you see? Positive biuret test. We can, for fun, try it for other things such as a bit of milk protein. What do you have in milk protein? What is the protein that you have in milk? Yeah, what do you have in milk? Casein. Casein protein? Yeah, that's the protein you have, right? Storage protein in milk. So nikan jolly rakalabalu. May I haven't done this actually, right? So let's see. I got some fat-free milk because that's going to isolate the protein. So, you only have proteins and lactose in this. Okay, sugar and proteins idhara idin. The fats have been removed. Okay, so let's give it a try. Well, people, you can see it's white colored. Taggala there was a purple color, right? Wow. I'll add. I can use a normal test tube because I'm not heating it, ne? Right? Boiling tubes use can on. Can you see? At the back, can you see? This is what? What was previously? This is just fat free milk, right? So, you do have some protein, eh? You can pass this. Bondipa. Okay. It has copper sulfate and sodium hydroxide. So, nikan jolly tinna gama me wage ingredient labels kiyawanna. What do you have? Okay. Per 100 milliliters, 3.4 grams of protein here. Okay. So, the advantage here is the total milk fat is 0, Maggie, right. So, for fun we can do the next test for full cream milk, we can do a lipid test. Okay. We can do a fat test for the full cream milk, the normal one, okay, right. So, let us see. So, what do we do? We have done 3 practicals, we have 2 small ones left, starch and lipids. Okay. More than break again. Yeah, small one, huh? Right. Okay. Online people, take a break. 15 minutes. Okay. We have to finish the practicals before the next class. Right. What about egg white? Right. Okay. Join at 5:45. Okay. Any questions? So if you like, you can download the practical guidelines. That is the sort of the official one. That is from the NIE, right? I think I had a PDF. Yeah, I'll send to your group as well, and online people, I am sending it to the group chat as well. If you want the official sort of the NIE practical guideline, right? Because I have exactly put it in the resource, sorry, in my tute. It's the exact version that is I'm giving you, right? Because you know I'm not going out, right? I might tell you things, extra things, but I'm not going to give you extra things to take home, right? Because at the end, while the learning can be interesting, the studying should be strictly from what you are recommended. So I can tell you things that are interesting in the class, but I have to stick to what you should be learning always. Okay. Too cold? No, right? Too cold? Okay. Tell me if you are too cold. Okay. Is it 23? No, right? 27? Okay. I need my glasses. Okay. Right. Everyone is not here yet, neither. So, clear about our bioret test? Yeah. So, we used two things, neither. One of the test kare egg white and sort of milk protein. Milk protein, what do you have in milk protein? Online people, yeah? Casein. Casein, no? Right? So, then, I mean, you know, there's separate whey proteins, right? There's a component called whey that is also, there's a sort of a group of amino acids that are found in proteins, sorry, in the milk that we use as supplement. But casein is a more stable protein that you can find in milk, right? So, casein is a bit difficult to digest. That's why these people, I mean, you know, you can take protein as supplements, protein supplements, then we usually use whey, the whey component of protein, sorry, milk as your sort of the dietary protein. We are whey protein gahanawa kiyala kiya. Yeah. So, that is, I mean, using the whole milk, right? Pure sort of whole milk, you can do several things. No? You can coagulate the lipid part using certain enzymes such as rennet, right? Renin wage enzyme එකක් දාලා you can coagulate it to the lipid part down to produce cheeses, cheese and butters, right? Then you can take the liquid part that is called the whey, right? When you extract all the proteins out of that, you have the whey protein powder. Okay, so those are the milk products that you usually have. Then what are you left with? Only lactose. Okay, to the lactose, what you can do is what? To the lactose, when you are making milk powder, you have a watery solution of lactose. That is when you are making milk powder. So, you spray that watery lactose over a sort of a high heated, there's a room, right? Vaporization room, you spray it over that room. So, you get a powder. That powder is not water soluble or doesn't taste good at all, right? So, you add to that certain lecithins and sort of palm oils to make it more soluble. Okay, then other flavorings, emulsifiers, or then you can have a nice milk powder, right? So, it doesn't have anything that is from the original milk except for a bit of lactose. Okay, none of the milk proteins, none of the milk fats, nothing. Okay, so that's the classical way. I don't know if these new full cream milk powders have a different way, but that's still the technique. Okay, you extract all the other things from milk, such as the milk fat for cheeses and butters, and for the milk proteins to make these whey protein products, and rest becomes milk powder. Okay, clear? Anyway, okay. Then sort of I wanted to talk a little bit about me. Sort of I talked about this for my 2027 people also, and 2026 people, they also complain of this lot. They say they can't focus. එක දිගට focus එක තියාගන්න බෑ when they are studying. their focus goes here and there, right? This happens mainly when they are doing bio, but then කියනවා physics ගානක් එහෙම click වෙන්නේ නැහැ කොච්චර වෙලා කල්පනා කරත්. දැන් ගොඩක් අය ගන්න එකක් තමයි imagine you have a sort of a chemistry ඉස්සරහට conversions එනවා, right? And then in physics you have mechanics complex questions. ඒ වෙනකොට නිකන් you have the sort of ගැම්ම තියෙනවා. හරි මේක කොහොම හරි මේ within පැයක් ගියත් කමක් නැහැ. මේක අද කොහොම හරි ගාන හදලම තමයි නැගිටින්නේ. කියලා ගැම්මට වැඩ කරනවා. වැඩ කරනවා කියන්නේ you try to do it. එහෙම මෙහෙම මේ equation එක දානවා, මේක දානවා, textbook එක බලනවා. You try to do it again and again and again. But there's a theory, sometimes solutions come to you when you let your mind wander කියලා. Okay? ඒ කියන්නේ so your brain start to make associations. ඒ කියන්නේ facts, theory paths that are here and there connect කරලා sort of a new approach එකකින් වට බලන්න පුළුවන් වෙනවා if you let your mind a bit more free. නිකන් එකපාරටම නිකන් අර ගැම්මට intensive focus එකෙන් කොහොම හරි මේක හදනවා කියලා ගාන දිහා බලන් හිටියා කියලා සමහරවිට එන්නේ නැහැ. I have tested it myself, physics. If I can't do a question, I just take a break. If I'm stuck in a question for more than maybe 30 minutes or 20 minutes, there's a theory that in this day and age, 20 minutes tamai our maximum sort of efficient focusing duration, right? Ethani ehata anyone, I mean many people tend to have their attention go here and there. Okay? So within 20 minutes, if you worked really hard, if you had a good focus, oh gana hadanna try kara, hadun naththam take a small break, 5 minutes, come back and try it again. Okay? පාඩම් කර ඉන්න ගමනුත් bio, right? For example, you are studying biological molecules. If you feel like after 20 minutes your attention is going here and there, මතක හිටින්නේ නැහැ, දැන් මේ පාඩම් කරපුවා දැන් මතක, take a break. Okay? Take a break and come back refreshed because your brain needs to have a bit of freedom as well. එහෙට මෙහෙට යන්න දෙන්න ඕනේ පොඩ්ඩක්. හැබැයි the tricky part is brain එක එහෙට මෙහෙට යන වෙලාවේ ඔගොල්ලෝ මොනවද කරන්නේ කියන එක. How do you let your brain wander? You have to train your brain to wander in a sort of a productive way. ඒ කියන්නේ දැන් ගිහිල්ලා sort of the main අර dopamine release එක එන්නේ මොකද්ද? එකපාරටම phone open එක නේ. Instagram or TikTok or whatever reels. Right? So that is not the way to let your brain wander. එතකොට you are sort of trashing your attention. Right? You have to find sort of productive ways. නිකන් සමහරුන්ට නිකන් ඔහේ නිකන් දවල් හීනයක් දකින්න එකත් that is a way of letting the brain wander. ජනෙලෙන් එළිය බලන් ඔහේ 5 minutes ඉන්නවා. Right? Maybe music without sort of visuals, just music. I had a sort of a thing, movie soundtracks අහනවා. Movie soundtracks අහනවා. Not songs. So there වචන දිනවනේ song වල. I tend to get distracted by songs which have words. ඒතකොට වචන එනවා ඔලුවට. Right? Songs with words. I listen to movie soundtracks. Like Harry Potter, Pirates of the Caribbean, Avengers, and they have a soundtrack. No? So I play one of those soundtracks, movie. Then I sort of get my brain refreshed and come back to work. Yeah? This is techniques. Okay? And then just go and take a walk. If you have a pet, right? Whatever. 5-10 minutes, take a break, but not usually devices. Go and talk to your mother. Right? These things will be very effective when you are close to the exam. Then 2026 sort of 4 months to the exam, gedara pisu natana, right? Ammara karadara karna, ballata karadara karna, motama karadara. That's good. That's good ways of refreshing. Okay? So I recommend that you don't use devices as the way of refreshing, and also raise a focus again, that's not going to be effective. Okay? We call it burnout. You are going to get burned out. That is not sustainable. So train yourself to let your brain wander when it needs to. Right? Give some breaks and make the work enjoyable. What's that? So bio test is done. Make another. Do this, right? Make another. Right. Let's come to practical experiment number 3, identifying starch in the lab. Identifying starch in the laboratory. Online people? Identifying starch in the laboratory. Very simple test. I'll just do it and show you. And so, right? So the component or the sort of the reagent, not a reagent, sort of chemical agent that we are going to use for this is iodine. Iodine in potassium iodide as in the form of potassium iodide, right? Iodine solution, right? What is the original color of this iodine? It is brown, okay, brown color. So we can do it in several ways, right? So one way you can do it is you can put some iodine to a piece of bread and try to see what's the color change. So this is iodine, okay. So it's usually brown color. It's unopened, okay, right? Okay, so I'll show you the original color of iodine first, right? So iodine looks like this. It is brownish. Can you see? This is the color of iodine, okay, brown color, right? Some still. Can you see? This is the color of iodine. I'll put a drop to this white tile. Okay, so it's like marmite colored. Okay, right. Now what we are going to do is we are going to put a bit of that iodine to this piece of bread to see if we can observe any color change. Okay, so this is the color of the bread. Okay, when you put iodine, okay, can you see? Black, dark blue, deep blue, black. Blue black color is the color change. Okay, can you see? Completely black, right? Brown iodine, black kuna, right? That is the color change, right? So this white refined bread mainly starch, no? Vena mukut naha, right? White bread wala vena mukut naha, nothing, no vitamins, no other sort of if you take brown bread, you have other vitamins and things, no, right? Then you have a bit of fiber. Aromeo mukut naha if you take a piece of white bread like this, okay? Completely starch. Then we can do this for some pure starch as well. We can pure starch, you can do this, okay. So this is palm piti, okay. So we take a bit of starch, that is your normal wheat flour, right? Okay. To that also, so this is the color, no? White. To that also you can add some iodine, okay, because it's not very, it's mixing very nicely, right? Can you see? It is reacting with the top part of it, right? Or we can do something different. What we can do is we can make a starch solution also, right? Starch solution nak hadala karannat puluwa, right? Shall we do it? I have a small one more. No, we'll make it to this, right? We'll make a small starch solution. Get a spoon somewhere. This spoon. Oh yeah. So starch, it doesn't dissolve very well, okay. So it doesn't dissolve very well. So it's going to be a sort of a colloidal solution. Okay. තලපයක් වගේ එකක් තමයි එන්නේ. Okay. Right. To that also, let's add some iodine in potassium iodide in the form of potassium iodide. Okay. So this is the white color iodine, sorry, white color starch. You have to add a bit more. how black it becomes. Yeah. Okay. That's a classical test used to identify any form of starch, right? Can be in the solution form or whatever. This is the color change you are going to observe. Okay, right. So Megam, I'll do the other experiment as well. Abhi mega QLM, then I'll give you a small note later, right? Identifying starch in the laboratory, practical number 3. Okay, identifying starch in the laboratory, practical number 3. Starch is slightly soluble in water in which it forms a colloidal suspension. It can be tested in suspension or as a solid. Okay, so this is the solid experiment that we did. Add 2 milliliters of 1% starch solution to a test tube, few drops of iodine in potassium iodide. A blue black color is observed. A blue black color is observed. Okay, pretty simple experiment. Okay, so that is the how you identify starch. Any questions? Abhi ඊළඟ experiment එකත් එහෙම ටක් ගාලා කරමු, right? There is identification of lipids. Okay, let's read that practical. Practical number 4, identification of lipids in lab using Sudan 3. Sudan 3 is a red fat soluble dye that is utilized in the identification of presence of lipids, triglycerides and lipoproteins. Okay. Sudan 3 reacts with lipids or triglycerides to give a red stain. So Sudan 3 is a non-polar dye. Okay, this is Sudan 3. It's a non-polar dye. It forms, it dissolves in anything non-polar. You know, lipids are non-polar. Lipids are non-polar. That's why it doesn't dissolve in polar water. Water is insoluble, right? So that's what we are going to test. So what we can do is, to a test tube, okay, you can add some පිරිසිදු පොල් තෙල්, right? අම්මගේ coconut oil, okay, right? So what we do is, let's make a sort of a double layered structure, right? Yeah, we'll add the coconut oil first just to see, right? What's going to happen? We'll add a few drops of coconut oil. Okay. Then to that we'll add some water. So Kianaban, who is going to float and who is going to be at the bottom? Water, no? Water is going to settle to the bottom because water is heavy, no? Water is heavier. Okay. So now we have a sort of a temporary suspension. The mega, it's going to get separated. Oil and water temporary suspension you have. Okay. So what you can do is, you can take some Sudan 3 and put a few drops there and see how the lipids can be identified. It's a non-polar dye, so it dissolves in anything only non-polar. Water is what? Polar. Because of what? Our hydrogen bonding, because of this uneven charge distribution between the negatively charged oxygen and the positively charged hydrogen, you have water molecule which is a polar molecule, right? So we take some Sudan 3 dye. You can see it's very red colored. So this is what? Our oil and water suspension. We add the Sudan 3. Okay. Can you see? The oil is on the top, right? So if we give it a shake, it's never mixing with the bottom part. Okay, let's keep it for a while to get settled, right? We'll keep it for a while to get settled. You are going to see how the oil and the water is separated. Okay, it never mixes with the water at the bottom. Can you see? Oil තියෙන්නේ උඩ, water at the bottom. So it mixes only with the oil. Got it? Can you see clearly the two layers? Okay. හලාගන්න එපා, right? 7:30. There's another class, right? I can do until 7. Yeah, sure. Okay. So that is the Sudan 3 for lipids. Any questions? No, right? So let's quickly take a note of those two experiments, and we can go to a bit of microscope. Any questions? So Benedict's is all stuff now, so I'll erase this side. Okay. So we actually use this starch and the iodine test in a upcoming practical that is in the same practical guide, that is enzyme activity. Okay, you will see there's another practical number 7, demonstrate enzyme activity and to determine the effect of temperature. For that practical also we use iodine solution. Okay, so that's how you get this basic knowledge to apply in later experiments. Okay, or to any substance that contains starch, if you add iodine in potassium iodide, you are going to get a blue black color. So starch identification, how do you do it? Two ml of starch solution to a test tube, 1% starch solution, right? So make you can be more elaborate and use potato extract only, you can get starch and you can do so many elaborate things, right? Because thing just you can dissolve a bit of wheat flour, you get a sort of a colloid, right? 2 ml of 1% starch. Okay, then what do? You add your, it's brown colored, I'll use the orange one, right? So I2 in Ki. Okay, mehema it is sort of presented to you, iodine I2 in Ki. Okay, I2 in Ki. What is going to be the observation? You are going to have a what? Blue black. Blue black color. Okay, quickly draw this. Page number 13, Taggalandin. Okay, so I'll erase this side also. Bioret test clear, right? I think I gave you a homework last week as well, right? Short note tell. Okay, we'll check it next week. Okay. So next week plan is going to be we are going to finish the nucleic acids with the end of RNA. RNA is a bit of a long story. Then what we are going to do is we are going to start the cellular structures, starting with a bit of history, and I mean I'm going to start the microscope part today as well. But let's see if we can finish it, right? If we can finish it, I don't think we will be able to. But let's take the introduction into microscopes at least. Then Sudan 3 for identification of lipids. So how do you do that? You take some oil into a test tube. I'll draw the oil into in this sort of orange color. Two milliliters of oil. Then what do? You add a similar volume of water to this, right? So what's going to happen when you add water to oil? The water is going to be at the bottom, oil is going to be at the top. Neither oil is going to be at the top. Okay. Equal volume of water. Right. Then what are you going to do? You are going to add a few drops of Sudan 3. Few drops of Sudan 3. So මොකද වෙන්නේ Sudan 3 add කරාම, right? What's going to happen? Water layer no issue at all, remain unchanged. But to the oil layer is going to change its color, ne? Red. Few drops of Sudan 3 and shake. Few drops of Sudan 3 and you shake well. Color of the water remains unchanged. Okay, quickly draw this. Nice if you can use some colors. Gedara gihilla if you can add some colors, that's fine. Okay. It's all color changes. Ne? Color changes are the things that you should retain in your brain. Okay, the color changes because that's what's questioned in the MCQs. Right, that's what's going to be questioned in MCQs. What is the color change? In future there can be structure. This is definitely in your school papers. You are going to have structure disease from practicals. Okay. So you can see I add a bit of too much oil, ne? Othurumadi, right? Online people, any questions? Any questions? Yeah. You have any questions? No. So that's all about practicals, right? So I'm going to give you a small paper. So cool down one next week regarding these practicals, because I mean today we won't be able to discuss much questions because of the other practical that we have to do. Okay, microscopes, right? Before that, how is the resource book situation? Resource books. We can move on to the second part of the seminar, that is from microscopes. Okay. Any questions up until now? No. Okay. So shall we go to your resource books? Resource book again. Okay. I think so. Last week in this class we started cellular basis, right? Cellular structure we started. Plasma membrane net kata karane. We drew the plasma membrane. Yeah. This is after New Year. This is the first, we are meeting, ne? Ah, ekai me mataka nahe, right? Matak mataka nahe. Okay. So take out the resource book. So then, because now you have three documents, තුනක් අතර switch වෙන්න වෙනවා. Okay, resource book, color tube, question tube. තුනයි තියෙන්නේ. Okay, you have to jump between three things. Okay, resource book is your reference. ඒක වෙනම තියාගන්න. The color tube is where you write down some short things, and question tube එකේ තමයි usually we do questions, but now here we have one more practical to discuss at the end. Okay. We put up microscopes. Okay, resource book, what's the page? Microscopes. So what you need to know is, if you take the history of biology, I mean for the development of or the progression of a certain science, being able to observe that science field is very important, right? For example, if you want to study cells, if you can see cells clearly, that is a big help. පේනකොට ඕක ස්ටඩි කරන්න හිතෙනවා. පේන නැති දෙයක් ස්ටඩි කරන්න අමාරුයි. Right? So, as I told you, microscopy and the study of cells has a very close relationship. Right? Cells පේන්න ගන්නකොට තමයි මේක ගැන ස්ටඩි කරන්න ඕනේ කියලා හිතෙන්නේ. Right? So, therefore we say development in cytology or the cell study and histology or study of tissues has a very close association with what? The development of microscope techniques, or the invention of microscopes basically, right? Microscope එක හැදුවාම cell biology and sort of the cytology sort of developed. Okay, so what is it, page number, what's the page number? Microscopes. Oh yeah, page number 22, ne? Yeah. Everyone has, right? Right. Okay, there's a hingayak තියෙනවා, resource books, because all the 2028 people are buying resource books in the kind of hingayak තියෙනවා, right? Every row has at least තියෙනවානේ. Okay, temporary එකක් තියෙනවා, right? So let's read. Advancement of the cytology is mostly based on microscopy. Okay, advancement of cytology is based on microscopy. Discovery and early study of cells progressed with the invention of the microscope. Sort of introduction things. Light microscope. Okay, so we are mainly focusing on light microscope and another form that is called electron microscope. Light and electron microscopy. We are going to mainly focus. Okay, so if we discuss about microscopes, microscope is an instrument that we use to magnify things, right? Magnify. The main function of a microscope is to create a bigger image. The may if I am doing a 10 times magnification, you are going to see an image 10 times. But is it a real image? For physics, real images versus virtual images. Okay, always usually the images we produce using microscopes are virtual. They are not real. You can't project them to a screen. Okay, so using microscopes, we are going to create an image that you can see in your eyes, a bigger version. Okay, so this is the object size. If I create something 10 times bigger of it, that is called the image, and there's a image to object ratio. අපි එහෙමයි microscopes වල power එක measure කරන්නේ, magnification power. Okay, so this magnification power is a very important property of any microscope, right? Whenever you are buying something like a car එකක් ගන්නවනම් engine capacity එක බලනවා, ඒ වගේ in a microscope the magnification power is a fundamental property. There's another property called resolution power that we'll talk. Okay, so microscopes are the microscopes. We have two main types to discuss. Before that, any microscope has three features. Any microscope has three features. Okay, I mean mainly it is relating to light microscopes, but okay, anyway, let's do a classification first. Classification microscopes in your syllabus you have two as electron. Okay, electron microscopes are there and light microscopes are there. So electron microscopes we'll talk about later. They have two types as transmission electron microscopes. We cannot TEM kela. Then there's another type called scanning. Transmission versus scanning. Okay, electron. Then what do you mean by this electron versus light? That is the medium that we are using to see the or visualize the object. In a light microscope like this, there's a way of providing light to whatever you're observing, and with your naked eye, because your eyes are what? They are photoreceptors, they are detectors for light. So you can detect light with your own naked eyes. So light microscopes also you can use your own built-in sensors. Right? But can you see electrons using your eyes? Eyes, the electrons? No. There's no sense organ in your body that you can detect electrons. Right? So for that, you need specific electron sensors that will be connected to a computer. Using the computer, using software, you can generate an image that you can see with your eyes. That's how electron microscopes. Pass. Light microscopes, you can have two main types. You can have simple microscope. What is a simple microscope? A single handheld lens. A single handheld lens. Convex lens. Single convex lens. Convex I think for O levels you had mirrors and everything, right? A single convex lens is a simple microscope. The sort of the most widely used one is the compound light microscope. Compound light microscope. So we are going to talk about compound light microscope a lot today. This is a compound light microscope. Okay. Okay. Online compound light microscope. Got it? right? So let's go back to the resource a bit. Resource again. So when I tell you to highlight, you highlight. Okay. Visible under light microscope, visible light is passed through the specimen and then through glass lenses. The lenses refract the light in such a way that the image of the specimen is magnified as it is projected into the eye. Image is magnified by bending light waves. Okay, because when light reaches this glass lens, I think you know this better than me, than physics, right? So whenever light reaches this, it is going to be bent, no? And bend on refraction. Okay, so because of that, it's like different mediums, right? Air to glass intervention, the light waves bend different speeds. Okay, so that is refraction. Highlight the lenses refract. That's very commonly questioned. The lenses refract in light, refract the light. Okay, in such a way that the image is about the specimen is magnified. Lenses refract. Okay. The simplest microscope is a single lens. Eka nikang underline kan. Okay, they can ask you what is a simple microscope. Okay. Compound light microscope. Compound light microscope. Okay, I'll we'll discuss about compound light microscope, the things in the resource book later. And come to the question tube. Question tube page number 14 of the question tube. Question number 14 of the question tube. Okay. Page number 14. Right. So practical number 6, identification of parts and functions of light microscope and the use of light microscope to observe specimens. Okay. So they say all light microscopes, what does it say? All light microscopes have three basic properties. All light microscopes have three basic properties. What are the three basic properties of a light microscope? Okay. They need to have these three things. Okay. Any light microscope, right? I mean, mainly for compound light microscope, right? They should have what? Lenses. Magnifying lenses should be there. Okay. Obviously, magnifying lenses should be there. Okay. That's how you bend. The lenses right? In a light microscope, what is the method of visualization? How are you seeing it? Right? You have to have a source of light, a source of illumination. A source of illumination, light should be there. Yeah, so if you take this microscope, you have lenses. This is an eyepiece lens. Okay, this is one set of lenses, and the other lens is this one. That's called the objective lens. Okay, so those are the lenses. What is the means of illumination or the source of illumination you can see in this microscope? Old fashioned it's a mirror, no? So using the light in the room, you can, it's a concave mirror. You can light around, it can focus into a point, concave mirror. Yeah? So using that mirror, you can focus light onto the specimen. If you take like modern labs, we in our pathology and histology labs, you have sort of digital lamp microscopes are there. You can control the amount of and the brightness of light. There are other things such as the diaphragm condenser to sort of control different properties of light. You can sort of make it become more pointy, sort of a point light. You can decrease and increase the illumination. So many things you can control of the source of illumination. But here there's nothing you can do, just a mirror. Finally, you have to have means of focusing, right? For different specimens that you observe here, you need to have different positions of lenses මේ lenses එකතන fix කරලා ඕන එකක් බලන්න බෑ, right? Whenever you are observing different things, you should be able to change the distance between lenses so you have a clear magnified image. That depends on the person who is watching as well or is observing as well, because each person has different eye properties. ළඟ පේන අය ඉන්නවා, දුර පේන අය ඉන්නවා, එක එක eyeball sizes තියෙන අය ඉන්නවා. eyeball size එකත් matters, okay, right? So because of those changing situations, you should be able to change the sort of the positions of lenses in the microscope. Can you see? That is the means of focusing. So this is called a focusing knob in the microscope. මේ දෙපැත්තේ කරකවන්න තියෙන focusing knob එක. Got it? So using the focusing knob, you can focus. Got it? Those are three properties of a microscope. ටක් ගාලා ලියා ගන්න එක. Page number 14. Online people, the tute was delivered, right? Most of the people, it was delivered to you. Okay. Then if you come to the other tute, make it out, put in, there's an introductory diagram there. Page number 1 of the diagram tute. So it shows why and how we utilize different microscopes in different situations. Okay. You can see at around 10 meters, 1 meters, 0.1 meters, 1 centimeter. They have found out our human eye is able to not, there's something called the resolution power. Our human eye is able to see up to a 1 millimeter level and 0.1 millimeter sometimes clearly. Okay. You can see the unaided eye කියන එක යනවනේ the product below 1 millimeter. That depends on person to person as well. So unaided eye stops there. The unaided eye stops there. Okay, right? Then yellow color part, that is usually where you have normal light microscopy, right? Where you have normal light microscopy from about 10 micrometers to about 100 micrometers in the 1 micrometer වගේ වෙනකම් you have usual light microscopes. Then so you can see most of the plant cells, animal cells, nucleus of cells, bacteria, mitochondria, even you can see. But if you want to see some viruses, small bacteria and very small subcellular part components such as ribosomes, cellular proteins, lipids, up to that level you can observe sort of big protein molecules using electron microscopes. Then if you want to observe the structure of other molecules and also if you want to look into the atom, you need other techniques, maybe X-ray crystallography, other techniques are there. Okay, so that is the end of biology, no? Right? Samane at the sort of organic molecular level, then biology, then it's sort of physics and sort of atomic physics and other things, right? So that's all we need to know at this point. Clear? So we can move on to the proper light microscope components. Proper light microscope components, we can move on. Any questions? So if you take a compound light microscope or even any microscope, there are two parameters that we sort of measure in any microscope, two parameters, those are your magnification and resolution power. No, I can still use that side. Okay, right? There are two parameters that we measure in a microscope. That is how big of an image it can generate. Okay, so that is called magnification. Magnification is the ratio of the image size to the object size. So you can say you can divide image size by the real object size, and you can get how much of a big image or like a bigger image you can create using a certain microscope. Okay, so come to the resource. There's a definition there. Resource again, there's a definition. Yeah, what does it say? Bottom of the page you have, right? Magnification is ratio of an object's image size to its actual size, right? Magnification is a ratio of an object's image size to its actual size. Okay, so the image of the object produced to its the object's actual size. That is what you mean by magnification. So if you take your light microscope, what is the maximum magnification? Light microscope, it's about 1,000 times. You can magnify about 1,000 times. Okay, if you take a light compound light microscope, I told you it has two lenses. I told you a compound light microscope. So I'll erase this part now. Okay, අපි මෙයා ගැන තමයි focus කරන්න යන්නේ. Okay, if you take a compound light microscope, I told you it's going to have two lenses. It's going to have two lenses, and those two lenses are called what? What are the two lenses? Yeah. So under physics world, you will have to draw these things called ray diagrams. Okay, ray diagrams and in right. Imagine this is the object. Imagine this is a bacterial cell that you are going to observe. Objecting. Okay, so the first lens that light from this object is going to go through is called objective lens. The lens that is closest to the specimen or the object. If you take a microscope, you mount the place of a bacterial cell on this stage. Light is coming from bottom. Mulimma, light goes through what? The specimen. Then it goes through what? This. That is the objective lens. So the first lens this light is going to go through is your objective lens. Then you have pattern, right? The light is going to first go through the objective lens. Okay. So make a complex ray diagram right? Right. You have to remember the ray diagrams for a compound light microscope you should be memorized. Telescope microscope and there's the near there are two versions for this. Okay, you should be able to remember. Okay, so a light wave bend there's another one that is going through the lens that is not going to be bent. I can't remember, right? Oh man, got it? And these two light waves are going to encounter the next lens. It's not physically that big in the to draw like a proper lens diagram. I'll make it a bit big. Okay. I can't exactly remember how these lens diagrams are, right? I am not doing physics. I have not studied physics for 5 years. Okay. Right. So these light waves are going to, light waves from what lens? This is the objective lens, is going to reach, make a, this is the objective lens. Okay, is going to go through the next lens, that is called the eyepiece lens. Eyepiece lens. May I again eyepiece lens. This one. Okay. At the eyepiece lens, it is going to be again magnified. Is going to be again magnified like this. Oh, bend oh, no, it's not going to be Omahari. It's going to be bent here like this. Omahari. There's going to be a magnified image produced upside down, sort of here. This is the magnified image. ඔයගොල්ලෝ පස්සේ මේක ඇඳලා හරියට ඇඳලා ඉල්ල මට කියන්න in like when you are doing light. ඔයගොල්ලෝ light කරන්න about maybe 1 year වත් ඇතිනේ, 8 months or 10 months. මොකද mechanics කරනවානේ මාස 6ක් විතර තිස්සේ. Right? Okay. So this is the image that is going to be produced at the end by these two lenses. Right? At the end by these two lenses, we are going to keep our eye. We are going to keep our eye here. Whether no mahathiela om balno. Okay, this is your eye, and you are going to see this image. This is the image. Then this is object. This is image magnified. This is how in a compound light microscope you produce a magnified image. Okay. Clear? Okay, right? So you can say, if you take a compound light microscope, you can write another equation. Okay, magnification is also equal to objective lens magnification multiplied by eyepiece lens magnification. Eyepiece lens magnification. Yeah? Then ekatuwak name. If you take a microscope, standard microscope, okay, standard microscope gatto, your eyepiece lenses are fixed. Their magnification is fixed. You can't change lens, same lens, right? So usually standard is eyepiece lens has a magnification of about only 10 times. Okay. Objective lenses, you have some options. They make it, right? Right? The objective lenses are only one is remaining. Usually objective lenses about 3 or 4 is there. Right? You can see the slots for the other objective lenses. Okay, usually about 3 or 4 objective lenses are there per light microscope, and you can switch between different objective lenses. This is called the rotating nose piece. Okay, using the rotating nose piece, you can switch between different objective lenses of different powers. Right? So usually we have 3 objective lenses. One of the options, there's a 10 times magnification one, there's a 40 times magnification one, there's a 100 time magnification. So you can see, when we combine, you are going to end up with this maximum magnification of a usual light microscope 1000 times. Right? Objective magnification multiplied by eyepiece magnification is the total magnification of a light microscope. Okay, all right, clear? So let's write this down. Here, okay, question tube, page number 14. Magnification is killer. There are some dotted lines. Magnification is killer dotted lines ටිකක් තියෙනවා. So what is magnification? Write down. is the ratio of an object's image size, is the ratio of an object's image size, write down, is the ratio of an object's image size to its actual size. අපිට මේ පැත්තේ යන්නත් පුළුවන්. Right, is the ratio of an object's image size to its actual size. Okay. Okay, then there's some space at the bottom. Write down that equation. This is not just magnification, it is a total magnification. Total magnification is equal to objective lens magnification multiplied by eyepiece lens magnification. Okay. Next, there's another definition or another parameter that we test. I mean, we need to know about a certain microscope. Microscope before using a certain microscope, you need to know the other parameter. It's not very commonly, I mean, because it's fixed for all light microscopes, right? Light microscopes, that is a fixed property. That is called the resolution power. Resolution power. Okay. What is the definition in the resource book? We can highlight. Okay. Minimum distance between 2 points that can be distinguished as separate points. Okay. Okay. Highlight the resolution power. It's a, as they say, it's a sort of a measurement of how clear the image is. Then using this ray diagram, you can see you can magnify the image to a very large size. Okay, but the problem with magnifying is there's going to be a certain point, there's a limiting factor. ඕනම දෙයකට ඕනම හොඳ දෙයක limit එකක් තියෙනවා, right? Okay, magnify කරගෙන ගිහිල්ලා there's going to be at a certain point, what is going to be the limitation is, yeah, in a light microscope, who are we using? Light is the electromagnetic spectrum is there, මේ පැත්තේ radio waves, ඊළඟට මොනවාද, microwaves, light, ultrasound, sorry, infrared, ඔහොම යනවනේ, right? Radio waves, yeah, ඊළඟට මොනවද තියෙන්නේ, microwaves, gamma, x-rays, blah blah, right? So you know light has a range of wavelengths, or you can say frequencies or wavelengths, right? So when you are keeping on magnifying an image that is produced by light at a certain point, because of the high wavelength, right, high wavelength of light, the clarity of the image is going to drop. Okay, what do you mean by the clarity? Then imagine you are you can see a shadow of my hand here, yeah. Why is that shadow produced? Because light coming from all these light sources are being stopped by my hand. Yeah? So you can see a sharp sort of deficiency of light. Light that's why you can see a shadow. Yeah. So you can see a sharp image because the light is not going around. The make waves physics. Right? When there's a barrier, waves, if the wavelengths are small, waves, light waves are coming like this. If there's a barrier, the barriers are going to stop the waves on the two sides, and the waves are going to go through the gaps. Take a through. Yeah. So this sharpness, the metana, you can see a image. The sharpness of that image or the clarity depends on the wavelength of the waves that are going and hitting the barrier. Okay. So light magnification, at a certain point, the wavelength of light is going to make the image blur. Low like a point the image is blurred. Okay. Right. So that's why we say resolution power inversely proportional to lambda. Lambda wavelength. Then inversely proportional to wavelength. Again, wavelength increase when resolution power goes down. Yeah. Then wavelength increases, resolution power goes down. Okay. To be honest, that's the exact reason why scientists had to discover or use electron microscopes, because electrons have a higher wavelength than light. So you can keep on magnifying using electrons. The light microscope large bacteria, nucleus, mitochondria. Right? If you want to see beyond that, you needed a source of light or sort of source of illumination with a low wavelength than light. That's why you had to invent the electron microscope. Okay. Write down in the question tube, resolution power is, write down the definition. Resolution power is the minimum distance. Resolution power is the minimum distance between 2 points. Resolution power is the minimum distance between 2 points that can be distinguished as separate points. Resolution power of the light microscope, this is the value. 0.2 micrometers. Beyond 0.2 micrometers, you cannot see clearly using light. Okay. Resolution power of light microscope is 0.2 micrometers. So beyond that, you can magnify, but no use. Pain, blood. Okay. Okay. Clear? Okay. Next up, we have to identify the parts of the microscope. Parts of the light microscope we have to identify. So now you know the theory, one of the lens they got, lens they combine to produce the total magnification like this, but the magnification is limited due to resolution. Okay, those kind of properties you know. Now we are going to see the lens they got high color. That's what the early microscope looked like. Then last week we talked about Robert Hooke. Yeah, what about Robert Hooke? So he had a sort of a basic tube beaker lens there, right? Okay, it didn't have most of the other things such as the ability to focus and everything else, right? So we are going to see if in a modern light microscope monad TN1 properties, right? Okay, right. So page number 15 of the question tube. Okay, you can see there's a list of parts. Okay, there's a list of parts. So we are going to write down the functions of each of those parts. So I'm going to show you the parts, and you are going to write down the function. Okay, then making ඇති parts මෙතන තියෙනවා because මම කියලා මේකට bind එපා. It's a simple microscope. Okay? But anyway, it's a binocular microscope. You have two eyepieces. Usually, most microscopes, I mean the school microscopes, සමන එක eyepiece එකයි නේ තියෙන්නේ. Using this binocular vision, you can have a sort of a depth idea, right? If certain cells are above this or beyond this, right? ඒ වගේ sort of a better sort of a 3D view you can get using if you look through two eyes. Okay. Okay. Come to parts of the microscope. Now මුලින්ම තියෙන්නේ main switch, right? So that is talking about a light sort of electric microscope, right? So the light source is electronic. Right down in front of the main switch, it's near simple, turns the light source on and off. Main switch turns the light source on and off. Right? Turns the light source on and off. Okay. Turns the light source on and off. Light intensity knob. I'll show you those. Okay. Light intensity knob is, I can't show you that here. If you have a sort of a bulb illuminated light source, there's a switch on this side that you can turn and decrease the brightness. Have you had in your house, you can decrease and increase the brightness. Then the lamps are there. Certain LED lamps, I think you still can, right? So illumination very you can control using the light intensity knob function. Adjust the light intensity. Adjusts the light intensity. Okay, adjust the light intensity. Okay. Then eyepiece, important part. Here is the important part. Yeah? Magnifies the image. Write down. Magnifies the image produced by the objective lens. Magnifies the image produced by the objective lens. You know. Second magnification. Magnifies the image produced by the objective lens. Okay. Field of view. Okay. You can write down. The circular illuminated area. Circular illuminated area seen through the eyepiece. Circular illuminated area seen through the eyepiece. Circular illuminated area seen through the eyepiece. Condenser. Okay, so condenser is another part that is missing here. Condenser, I mean, it actually has another set of lenses, right? Using the condenser, you can do a bit of focusing of the light source. Then light source, imagine from the light source, you have light coming like this now, parallel enona. There's a condenser lens using the condenser, you can focus the light into a sort of a pinpoint. A point, right? Gala, you can sort of converge the light to a specific point on the specimen. Okay. Condenser, focus light onto the specimen. Focus light onto the specimen. Focus light onto the specimen. Diaphragm knob. එතන knob කියන එක කපන්න. Diaphragm. Okay. Diaphragm is the main part. ඒකේ knob එක ඉතින් ඒක control කරන එක. Okay. Diaphragm. controls මෙහෙම ලියන්න පුළුවන්. So what is the diaphragm is, you can use the diaphragm. It's not very commonly found. It's like අර මෙහෙම කැමරා වල දැකලා තියෙනවද? There's a hole you can open and close. ඒ වගේ ඇරනවා වහෙනවා. Have you seen all digital cameras? Click ගාලා ඇරලා වෙන්නේ. So the diaphragm is something like this. There's a switch, sort of a rotating මෙහෙම උස්සන්න පහත් කරන්න පුළුවන් කැලලක් තියෙනවා. Right? Using that you can open and close. Okay? Right? That is usually found in sort of these sort of mirror illuminated lamps. Or the light you can't control, right? Because unlike a sort of a electrical lamp, you can't control the brightness with a mirror. So you can control the light by opening and closing this sort of opening. Okay, so you can say diaphragm controls the amount of light reaching the specimen. Controls the amount of light reaching the specimen. Controls the amount of light reaching the specimen. Then stage, very very important part. This is the stage. At the back, can you see the stage? Within this glass surface is where you mount your specimen to be observed. Okay, write down the function. Stage, to mount the specimen for observation. 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 Okay, stage. Then specimen holder, that's not the common name. Specimen holder එකට අපි කියනවා the common name you give for it is stage clips. එතෙන් ලියා ගන්න එහෙම dash එකක් දාලා stage clips. Okay, stage clips. Specimen holder, more commonly stage clips. Okay, stage clips. Online people not sleeping, right? Okay, stage clips hold the specimen on the stage. Hold the specimen on the stage. Hold the specimen on the stage. These are the stage clips. Can you see? There's two stage clips, right? So you can move them like this, and a glass slide you can mount on top of it. Okay, have you seen glass slides? Physics class again, they might have handed over to be measured using the micrometer screw gauge, right? Thickness measure karone micrometer again. Yeah, or was it vernier caliper? Both. Usually micrometer again. The glass slide thickness measure. No, spherometer. Spherometer again thickness measure again. You can use the spherometer, right, to measure sort of a height, small heights measure. Anyway, okay. So stage clips, there is a function. Then revolving nose piece. Okay, revolving nose piece. This is the nose piece. When make nose piece, that is okay. That holds all these objective lenses. Okay. Okay, allows changing of objective lenses. Write down. Allows changing of objective lenses. Allows changing of objective lenses of different magnifications. Of different magnifications. Of different magnifications. Okay. Next, objectives. There can be 4x, 10x, 40x, 100x, and oil. Right? Oil I removed. Right? 4x, 10x, and 100x. Right? So 4x. Yeah, this is a 3x one. Right? Okay. So objective lens. What is the purpose? Write down. Okay. Magnifies the specimen for the first time. Magnifies the specimen for the first time. Magnifies the specimen for the first time. Next, right, then we have focus adjustment knobs. Focus adjustment knobs, those are important. Right, then if you take this microscope, there's only one knob. Ek knob එකයි තියෙන්නේ. දෙපැත්ත තිබ්බට that is the same knob. Okay, if you take a microscope, there are two ways you can focus this. There's two knobs. The first one is called the cos focus. Okay, අපි ඒක ලියමු function එක. Cos focus. Okay, you can say moves the lenses distances, moves the lenses large distances for rough focusing. Moves the lenses large distances for rough focusing. Can you understand, right? Whenever you mount something for the first time here, use this cos knob. That is going to bubugala, you can change, right? The distance between the specimen and the lens you can change quickly using the cos knob. Okay, so that is going to give you like a hari mulimma mata niyan lawata pena position eකට you bring using the cos adjustment knob. Okay, then what's going to happen is you can lock this cos knob, right? In certain advanced microscopes, if you want to use the next knob, that is the fine knob, you have to lock the cos knob. Then the cos knob you can't rotate. Samarla, you can't rotate the fine knob until you lock the cos one. Okay, so it's like two different. There are cogs inside. That is all the thing, right? So the cos one has like bigger sort of cog wheels, I think. So it moves the lenses faster. So this has only, I think, a cos one. Okay, fine one you can't even see. The lenses are not going, but it's adjusting them very slowly. Okay, right, write down the function of the fine focusing knob. For precise focusing of the final image. For precise focusing of the final image. For precise focusing of the final image. Anything you missed? Anything else? Fine focus and close focus. Sorry, we have 2-3 minutes. Next week the plan is going to be like this. We are going to draw a diagram of the microscope in the next page. We are going to label all these parts, and we are going to discuss the steps to observe this basic specimen. There is an onion peel. Okay, onion peel like observe karnati. We are going to write down the steps. Okay. Okay. And next week, okay, next week there's going to be a small paper that is our usual cool down එකක් වගේ එකක් will be there from microscopes as well as the practicals. නැත්නම් ඒක ඔය නිකන් කළා මතක වෙලා යනවා right පොඩි paper එකක් කළා ඒක නිකන් කොටලම දාමු right anyway so hari quick summary මොනවද අපි අද කරේ so we started with you are reducing sugar test yeah that is this නේද glucose වලට you put benedict and heat there's going to be a color change from the blue green yellow orange until there's a precipitate at the bottom right so with further oxidation there's going to be different colors so it has changed the color so you need to only remember the sequence of color changes මතකයි non reducing now what is the steps you add a base weak acid then you boil then you neutralize then you do the benedict test it's going to be positive okay then for starch come on starch somewhere at the back right what is going to be the color change the yellow brown iodine is going to become blue black lipids when you do the lipid test when you add a non-polar dye to any lipid it's going to only dissolve in the non-polar lipid okay Sudan 3 test biuret test I told you it's going to change color right originally what should it be if there are peptide bonds it's going to be a purple color that's it then we discuss the parts of the microscope so next week we are going to draw a microscope when we are going to do questions okay you can go okay hope you learned something and hope you can remember the practicals yeah 2 years and 3 months from now if you can remember then that's all it needs okay you can go online people any questions